Hi everyone. In this video, we will learn a recombination model, which is Holliday model of recombination. Before we learn Holliday model of recombination, let's understand what is genetic recombination. Recombination is the exchange of genetic information between DNA molecules. All DNA is recombinant DNA. Genetic exchange works constantly to blend and rearrange chromosomes, most obviously during meiosis, when homologous chromosomes pair prior to the first nuclear division. During this pairing, genetic exchange between the chromosome occurs, and this exchange, which is crossing over, is one of the results of homologous recombination. This recombination involves the physical exchange of DNA sequences between the chromosomes. The frequency of this recombination or crossing over between two genes on the same chromosome depends on the physical distance between these genes, with long distances giving the highest frequencies of exchange. This recombination is an essential cellular process catalyzed by enzymes synthesized and regulated for this purpose. Besides providing genetic variation, recombination allows cells to retrieve sequences which are lost through DNA damage by replacing the damaged section with an undamaged DNA strand from a homologous chromosome. Recombination also provides a mechanism to restart damaged replication forks. Furthermore, there are special types of recombination that regulate the expression of some genes by switching the specific segments within the chromosome. Cells can put dormant genes into sites where they may get expressed. Now, when we talk about models of homologous recombination, before we do double strand break repair model, which will be the next video or recording, in this video, we will learn Holliday model. Holliday model is a very important model in homologous recombination. And although it's now clear that most recombination events involve some new DNA synthesis, a feature which is absent from this model, still the Holliday model very well illustrates the steps of the recombination, such as strand invasion, branch migration, and the Holliday junction resolution. Let's see what happens as per the Holliday model for homologous recombination. The first step is alignment of two homologous DNA molecules. What do you mean by homologous DNA molecules? Of course, we are learning homologous recombination. Homologous DNA molecules are DNA sequences which are identical to each other or nearly identical. When I say they are exactly identical, you would say then there might not be an impact or an outcome of a recombination event, and how will the variation or the genetic variation would result. But when we say identical, they are almost identical or nearly identical, but they are not exactly 100% similar. Despite this high degree of similarity, they may have small regions of sequence difference, and these different sequence variants are known as alleles. We know that alleles are the different forms of a gene. Even in Mendelian genetics, if we see a gene for the, I would say, seed shape, the smooth seed is result of an allele, and the wrinkled seed is result of a different allele. So alleles are different forms of a gene, and these alleles may have slight, I would say, sequence differences, and sometimes the sequence difference may be a one base pair. So in this case, you can see these are two homologous DNA molecules which are almost identical in sequences, and there are different alleles. The DNA which is shown in gray, if we see here, one second. DNA which is shown in gray here, their alleles are capital A, capital B, capital C. Whereas if we see that DNA, the other DNA molecule, which you can see here, which is shown in blue. it is small a, small b, small c. After the alignment, the next step is single-stranded breaks in each duplex. You can see there's a single-stranded break in this DNA molecule, and there's a single-stranded break in this DNA molecule. So in both the DNA molecules which have been aligned, there are single-stranded breaks. These single-stranded breaks occur at the exact position or the same position, which you can see in this step. And also why they are called as single-stranded breaks, because the break doesn't happen in both the strands. You can see that this strand, what you have here, is intact. And for this DNA molecule, you can see that this strand is intact. So the break occurs in only one of the strands of the DNA molecule, and it happens at the same position in both the homologous chromosomes. So after that, what happens? The chromosomes have aligned, single-stranded breaks have occurred in each of the duplexes. Then the next step is strand invasion. What do you mean by strand invasion? See, after the single-stranded breaks here in this step, there would be formation of short regions of base pairing between these two recombining DNA molecules. These pairing occurs when a single-stranded region of DNA originating from one parental molecule pairs with its complementary strand in the homologous duplex DNA molecule. So if, let's say, after the break, you have this end of this DNA strand in this DNA molecule. Now this is gonna pair, you can see, with the strand of the other DNA molecule. So you can see how this is pairing with this. And let's say this end is gonna pair with the strand of the again other chromosome, which you can see here. So this is called a strand invasion, and the two DNA molecules are connected by crossing DNA strands and the X-shaped structure that you can see here. This X-shaped structure is called as a Holliday junction. After that, what is going to happen? There would be movement of the Holliday junction. So at this step, you can see this X-shaped structure is at this point. Now in the next step, you can see the X-shaped structure has moved here. So how this Holliday junction can move, or what happens if the Holliday junction moves? The Holliday junction moves along the DNA by the repeated melting and the formation of base pairs. When the junction moves, base pairs are broken in the parental DNA molecules. So at this point, you can see, now we know that the DNA is not colored, but for the explanation purpose, they have shown one DNA molecule in gray color and the other DNA molecule, which is a chromosome and which is homologous to the other chromosome, blue in color. So in the gray DNA molecule, the two strands are pairing with each other, of course, with the hydrogen bonds between the nitrogen bases. Now, these two strands are peeled apart. This gray strand would be detached from the other gray strand. These strands are peeled apart, and this is melting. That is, the hydrogen bonds are broken between the nitrogen bases, the strands are separated, and this is melting of the DNA. Why this melting occurs? So that this gray strand can pair with its complementary strand of the other DNA, which is the blue strand in the picture, what you can see here. So base pairs are broken in the parental DNA molecules, that is between this gray strand and between the blue strand and blue strand, while the identical base pairs are formed in the recombination intermediate, where you can see the gray strand is pairing with the blue strand, and here also the gray strand is pairing with the blue strand. This process is called as branch migration. After that, what happens? This Holliday junction has to be cleaved, and finally the DNA molecules have to be separated. Before we see the separation, they also say, since these two molecules were homologous, but they were nearly identical, but not exactly identical, there were, let's say, some sequence differences which may be of at least one base pair. So there might be some mismatches here, which you can see here. For the resolution, how the resolution can take place, and more important is that what will be the outcome or the impact of the resolution. Resolution occurs in one of the two ways, and it gives rise to two distinct classes of DNA products. Now, to make this cut size easier to visualize, the Holliday junction, which you can see in this step D, the Holliday junction is rotated to give a square planar structure here. So what you see in step D here, okay, which is this, it is same in the step on this slide. Only thing is it's rotated to give a square planar structure with no crossing strands. How the cleavage can occur, or how these DNA molecules or the Holliday junction can be resolved. You can see here there is site 1 and there is site 2. So the cut sites marked 1. Now when you see mark 1, after you have the square planar structure, when you see here site 1 or marked 1, the cut sites marked 1 occur in the two DNA strands. Now you can see this dotted line here. Now this dotted line is going through two DNA strands, not all the 4 strands. Because if you see this Holliday junction have two DNA molecules interconnected. If I say two DNA molecules interconnected, that means there are 4 strands. But if you see this dotted line here, the dotted line is passing through the two of the strands, site 1. So the cut sites marked 1 occur in the two DNA strands that were not broken during the initiation reaction. So I go back or take you back to step A, when the alignment took place, and when after the alignment the single-stranded breaks were made in both the DNA molecules. In this case, you can see the single-strand break was made in this strand, and you can see the single-strand break was made in this strand, in this DNA molecule. But the other strands are intact. The strand which is intact is one which I'm highlighting in yellow, and the strand which is intact here, the one which I'm highlighting in yellow. So when I say, coming back to the current slide, site 1, the cut sites marked 1 occur in two DNA strands that were not broken during the initiation reaction. If these strands are now cut and covalently joined by DNA ligase, the resulting DNA molecules will have the structure and the sequence that is the splice or the crossover products. This is called as splice recombination products or crossover products, because the original duplexes are now spliced together such that the regions from the parental DNA molecules are covalently joined together by a region of hybrid duplex. So this generation of splice products results in reassortment of genes that flank the site of recombination. So if you see here in the splice or the crossover products, of course, it started with two homologous chromosomes in step number A. After they were aligned, nicks were made, strand invasion took place, Holliday junction was formed, branch migration took place, and now in the resolution, again, this structure would be resolved to give two DNA chromosomes or DNA molecules. But the result is that you can see here that you have a capital A allele, but along with it in this DNA, you have small c allele. If I take you back to the first step when these two DNA molecules were aligned, you can see one of the DNA molecules, which is highlighted gray in color, has all dominant alleles, capital A, capital B, capital C. And the other one has all recessive alleles, you can say, small a, small b, and small c. But now at the end of the recombination, the result is that the DNA which has capital A, uske saath mein small c allele has come. And in between you have a heteroduplex DNA. And the other DNA molecule, you have small a, and along that capital C has come. And you have a heteroduplex DNA. So this is reassortment of genes, and these are crossover products or splice products, and it has a massive outcome. And this happens when the cut is made in the vertical plane, you can see, or in the strands which were not broken during the initiation reaction. On the other hand, is there any other way? Yes. The cut can also be made this way. So when the cut is made this way, again, you can see only 2 strands are involved and not 4. And I can call this cut as a horizontal cut. Here, the cut is made in the 2 DNA strands that were broken to initiate recombination. So if I take you to the slide 1, the strands which were broken, that means the strands in which the cuts were made. I'm talking about these strands in which the cuts were made. So if now the cut is made in the strands that were broken to initiate recombination, you have patch products or non-crossover products. And in this patch or non-crossover products, there is no crossing over patch or non-crossover products, and there's no reassortment, because if we see here, you have, along with the capital A, you have capital C only, and along with the small a, you have small c only. Of course, there's a heteroduplex DNA, but the impact is not as big as what you have seen in the other case. So whether a vertical cut would be made or a horizontal cut would be made, that will decide whether splice products will be made or patch products will be made. The same Holliday model, this is from the reference book, Molecular Biology of the Gene by James Watson. The same diagram is from other book as well in the PPT, which is from Benjamin Pierce. And you can see this is the similar thing. Diagram is from a different book, but the process remains the same, that the 2 double-stranded DNA molecules from homologous chromosomes align. How you can see a single-stranded breaks occur in the same position on both DNA molecules. Then the free end of each broken strand migrates or invades the other DNA molecule. Each invading strand joins the broken end of the other molecule, creating a Holliday junction, which is this X-shaped structure, and begins to displace the original or complementary strand. How the branch migration takes place as the 2 nucleotide strands exchange positions because of continuous melting and joining of the strands. And we can view this structure with the ends of the 2 interconnected duplexes pulled away from one another, square planar structure, rotation of the bottom half of the structure. And as we have seen in the previous diagram as well, that if the cleavage is in the horizontal plane, and rejoining of the nucleotide strands, you're going to see non-crossover recombinants or the patch products, where you can see along with capital A, there's capital B only, and along with small a, there's small b only. So if there's cleavage in the horizontal plane and rejoining of the nucleotide strands, that produces non-crossover recombinants consisting of 2 heteroduplex molecules. On the other hand, if the cleavage takes place in the vertical plane and rejoining of the nucleotide strands, you get crossover recombinants consisting of 2 heteroduplex molecules. So the Holliday model predicts non-crossover or crossover recombinant DNA depending on whether cleavage is in the horizontal or the vertical plane. So we have learned what is recombination, what is its significance, and the steps in the Holliday model for homologous recombination in this video. In the next recording, we are going to see the double strand break repair model.